Yeah. Hello, everybody, and welcome to the webinar on Paving the Way for Continental Scale Biology, Connecting Research Across Scales. I'd like to welcome all of those of you who may be listening in, as well as our speakers and our committee this afternoon. I'd also like to extend my thanks to the staff of the Board on Environmental Studies and Toxicology, which I direct, and the Board on Life Sciences, our collaborator in this study. On behalf of the National Academies, to welcome you all, and thank you very much to the committee and for the panelists this afternoon. I'm going to now turn the microphone over to our chair, Dr. Jack Liu, for some introductory remarks.
Thank you so much, Cliff. Hello, everyone. I would like to welcome you to the first of the 3 public information gathering sessions, Paving the Way for Continental Scale Biology, Connecting Research Across Scale. This is organized under the auspices of the Consensus Study Committee on Research at a Multiple Scale, a vision for continental scale biology. My name is Jack Liu, committee chair. And next slide, please. I will begin to acknowledge the wonderful committee members for the great help in planning this webinar. The committee's work is being conducted under the auspices of the National Academies of Sciences, Engineering, and Medicine in response to a request from the National Science Foundation. The committee's statement of task is available on the website of the National Academies, and the public can provide feedback on the project at any time. Next slide, please. I also like to acknowledge the outstanding staff team at the National Academies for the fantastic help in planning this webinar. The study is a collaborative effort between the Board on Environmental Studies and Toxicology and the Board on Life Sciences. Cliff Duke, who you just heard from earlier, is the study director. Tricia has been leading the organizing of this first public information gathering. Kat and other staff members have been very helpful in many ways. Next slide, please. Today, the committee is fortunate to be hearing several presentations from leading researchers working across biological scales. Following the keynote presentation and other presentations, we will take questions from both the committee and the participants joining us via live stream. If you are a committee member or a speaker, please use the raise hand feature in Zoom to ask your question. If you are an audience member joining us via live stream, please submit questions through Slido. You can upvote the questions from the audience you like to hear most. We encourage questions from the audience, but questions from the committee will be prioritized. Next slide, please. After the meeting today, anyone wish to submit written comments or other materials that are relevant to our charge should contact Cliff, the study director, or provide input and feedback through the project website portal. I also would like to emphasize this is a information gathering session. The comments made by individuals should not be interpreted as positions of the committee or of the National Academies. I want to note that this entire session is on the record and is being recorded. Now, it's my great pleasure to introduce our first keynote speaker, Dr. Janet Franklin. Janet is an endowed Campanile Foundation Professor in the Department of Geography, San Diego State University. She is previously a distinguished professor in the Department of Botany, University of California at Riverside from 2017 to 2022. A Regents Professor in the School of Geographical Sciences and Urban Planning, as well as a distinguished sustainability scientist at Arizona State University, where she was appointed in 2009. From 1988 to 2009, she was on the Faculty of Geography and Biology at San Diego State University. She received her PhD in geography from University of California, Santa Barbara in 1988. Her research expertise is in landscape ecology, global change biology, conservation biogeography, and geospatial science. Janet's scholarship seeks to understand the patterns and dynamics of terrestrial plant communities at the landscape scales. Her work addresses the impact of human-caused landscape changes on the environment and the biological diversity. Her outstanding accomplishments have been widely recognized. For example, she is a member of the U.S. National Academies of Sciences and a fellow of the American Academy of Arts and Sciences, the American Association for Advancement of Science, and the Ecological Society of America. She has also received many prestigious awards, such as the Distinguished Landscape Ecology Award from the North America Regional Association, the International Association for Landscape Ecology. Now let's welcome Janet. Janet, please take away. Thank you.
Well, thank you, Jack, for that very kind introduction. And I want to thank Jack and the committee for inviting me. I'm honored to be part of this information gathering session in support of the consensus study. I'm also thrilled to share the virtual, share the virtual stage with some excellent panelists today. And so I'd like to go ahead and get started so we can hear from them as well. Can see my slides? Can everyone see my slides?
Yeah,
okay, I see heads nodding yes.
Thank
All right, so let me start with this beautiful satellite image showing the Kumeyaay homeland centered on the Gold Star, where I've had the privilege to live, work, and raise my children, and now my grandchild, for which I am grateful. So I worry about two things, global climate change and biodiversity loss. This is perhaps not surprising because my entire life, certainly my career, has taken place during what's come to be called the period of the Great Acceleration of the Anthropocene, since about 1950, when all of these Earth system trends, as well as socioeconomic trends, have accelerated. And I'm concerned about climate change impacts on biodiversity, but not just that. I'm concerned about how multiple global change drivers impact biodiversity and how they interact with each other, in particular land use change. And I do study these problems at larger spatial scales. So I want to start by confessing that I'm a little bit uncomfortable with the term continental scale because it doesn't necessarily correspond to the ecological and biological hierarchies that I've been taught. And also it's a little bit terrestrial biased. I mean, why not ocean basin scale? And I say that not as a marine scientist, but as a terrestrial scientist who has worked in regions that were not continents. But we're not going to talk about any of that work today. So let's turn to this sort of foundational paper. These authors, in thinking about what do we mean by continental scale, these authors defined macro scale as regional to continental extents with distances spanning hundreds to thousands of kilometers, i.e. larger than landscapes, with biological, geophysical, and social components. So I think of that as sort of as the eco-regional scale, for example, in this classic sort of textbook. It's a hierarchy of spatial and temporal scales from landscape ecology, or perhaps as functional biomes, as a bunch of us recently defined in this classification with David Keith, or maybe vegetation formations, or provinces, floristic provinces, again, from these sort of classic examples. So at any rate, as a biogeographer, the large unit of biology that's useful when thinking about global change and biodiversity is the abstraction of the species range. And as a landscape ecologist, all my work links biological, ecological phenomena across scales. And so what I want to do today is share some examples of these linkages across scales. I'm going to talk about 3 examples. One scaling from genes up to species ranges, one from individual organisms to the species range, and then from species ranges to populations and communities. And then I'll close with a kind of a follow-on comment about an important component to linking those scales is, for me, is characterizing the biophysical environment experienced by organisms as a function of scaling interactions, particularly of the climate and the land. So let's start with this, my first example, linking scales from genes to species ranges, or from molecules to landscapes, as these authors said. Now, this is not my work, and so I'm going to stick pretty closely to my notes here so that I try to represent what they did accurately. This is a cool study by Fitzpatrick and Keller, which used population level information on genetic composition that was represented by SNPs, singular nucleotide polymorphisms, across the entire range of a species, balsam poplar, which happens to be kind of has a continental scale distribution spanning boreal North America. And they treated the genetic composition within the species as an ecologist would species composition in a community, modeling allele turnover, changing in composition as a function of macroclimate. And so in the first 2 maps, the colors represent the projections from their model of areas with similar colors would be expected to harbor populations with similar genetic composition. The top map is the background genetic spatial structure for the species, represented by hundreds of reference SNPs across the genome. The second map shows the genetic composition for the circadian clock gene, GI5, based on 22 SNPs, that is strongly related to turnover, and that composition is strongly related to temperature, which is what these little arrows with the strange names are supposed to represent, is bioclimatic temperature variables. And this gene is a strong candidate for local adaptation, possibly reflecting adaptive response of poplar bud phenology to temperature. And in the third map on the bottom, they then predicted the mean genetic offset for GI5 for this circadian clock gene genetic offset showing the place where gene-environment relationships will be most disrupted under future climate scenarios. So they did that for 9 climate scenarios for the mid-century. So locations with large predicted genetic offset, the warm colors in the map, occur in portions of the temperature gradient where turnover functions indicate rapid change in allele frequencies. And future changes in temperature are predicted to produce large predicted genetic offset. Okay, so this is a really nice example. It's not, there's been other examples of this work more recently. This was just one of the first ones I read. There's been, not surprisingly, some recent work on Arabidopsis thaliana by Moises Esposito Alonso and colleagues in Europe. But let's move on to the second example, which is something I'm more comfortable with because it's my own work. And that is looking at the effects of global change drivers on species distributions, and again, forecasting the future. So we're working down here in the realm of models. And again, I'm interested in the interactions among different global change drivers. So this example, scaling from species to species ranges, is from our work in the California floristic province, shown here. That's the Mediterranean portion, climate portion of California, excluding the Great Basin and the desert, Southwest deserts. It's the portion of California and adjacent regions that experience a Mediterranean climate with winter precipitation that we've had a lot of this year, and warm, dry summers. This area also has extremely high plant diversity and endemism, with something like 30 or 40% of the plant taxa in the floristic province being endemic. And so global change drivers in this region include land use change and also altered natural disturbance regimes, fire regimes, and climate change can interact with those other drivers as well. So the water challenged climate of California is projected under climate change scenarios to get warmer and effectively drier. So this is shown here, with the big map in the middle showing a climatic water balance metric, the climatic water deficit, which is the difference between the potential evapotranspiration and actual evapotranspiration. So higher values mean a greater water deficit or less water available for plant growth. And then the small maps show under 2 different climate models in the rows and 2 different emission scenarios in the columns, projected end of the century change. It's a difference in climatic water deficit. So the areas that get redder are predicted to have a greater climatic water deficit under climate change scenarios. So you can see, particularly in the high emission scenarios, a lot of the map gets red. The approach we use for scaling from individual species is to use species distribution modeling, also called habitat suitability modeling or environmental niche modeling, which is shown here, where values of climate variables as well as other environmental variables at species localities are projected into environmental space. This is shown with winter precipitation and temperature variable here. The colored dots represent the species occurrence and the gray dots the environmental background. Those relationships are then modeled using statistical learning methods and used to predict habitat suitability. Under current climate conditions, which these predictions can then be adjusted for current land use, removing areas of habitat that are not suitable due to land use. We then can calculate, and this is just an example for an endemic oak species, interior live oak. We then can calculate, among other things, we can make future projections of future habitat suitability under climate change scenarios, as well as under land use change scenarios. And then we can calculate based on that species exposure to climate change, which in this example, we're defining as habitat suitability change within the species current range. We did that over for many plant species and can then look for some generalities, such as how spatial range traits influence exposure to climate change, where we're defining exposure as the difference between current and future habitat suitability as a proportional change. And this can be summed over the entire currently occupied range of a species. So this is work by my PhD student, Brooke Rose, that was just published. And so I will briefly summarize what she found for over 100 species representing the California floristic province, was that those shown here in this regression tree analysis, those with higher exposure were species that had smaller range sizes, lower topographic heterogeneity, occurred at higher elevation, where there's no place to go but up with climate change scenarios, and fewer habitat patches. So small range size and few habitat patches, of course, being sort of classic attributes of risk to biodiversity. And vice versa for species with lower exposure to climate change. This can be contrasted then with what Brooke found for land use change exposure when we held climate constant and just did projections under using existing projections of land use change. In this case, species with higher exposure were those that occurred at low elevation, low topographic heterogeneity, so the places that are likely to be developed, and was less dependent on range size. All right, so that was my second example. My third example, linking species ranges to population and communities, comes from a macrosystems ecology program grant that was to Frank Davis and others. He was the PI at UCSB, UC Santa Barbara. This was during the first call from that NSF program. And so this project started back in 2011 and ended a few years ago. And in that project, we were able to characterize the climatic niche of the seedling stage for several tree species using a common garden experiment where we had 24 gardens across 6 sites across 4 sites spanning the southern portion of California. So in this experiment, we measured seedling survival as a function of bioclimatic variables. And so one of those results is shown here. So for a species of oak, this is showing survival as a function of 2 climate variables, the maximum temperature of the warmest month and the climatic water deficit, showing highest in 2 different years, or the 2 different colors. And this is showing the highest survival at low climatic water deficit and less hot summer temperatures, and in the hottest, driest sites, very low seedling survival, even for these hardy oaks. We could then take a model like this and use it to predict to the landscape to show the current distribution under the current year's climate of probability of seedling establishment or suitability of the landscape for seedling establishment. So in the map, that's shown as the greener areas of higher probability. The purple areas show the current distribution of adults of this particular species of blue oak. We could then use the same model and predict forward year by year using climate change projections. What we can see is that on the graph is that but towards the end of the century, the overall proportion of the landscape is that's suitable for seedling establishment is predicted to decline. But we do see these temporal good years, windows of opportunity for seedling establishment. However, the frequency of those and their magnitude also is predicted under this particular climate change scenario to decline towards the end of the 21st century. So so the temporal scaling and as well as sort of the life stage scaling was important. Indeed, our colleague Pep Sarah Diaz found that if you use 30-year climate normals or climate averages to predict seedling establishment suitability in this particular landscape, that you would miss a lot of the temporal opportunities. So only the blue areas of the landscape are predicted as suitable for seedling establishment based on 30-year averages, whereas using smaller time windows to permit establishment of seedlings during that time span would add the red and gold areas to the landscape as a window of opportunity, at least during some portion of the time span. Okay, then, so we could then link these distribution models for seedling establishment to BioMove, which is a population model that also includes some community interactions. This is a simulation model where species population dynamics are simulated through matrix modeling that includes scaling demographic rates by climatic suitability. Dispersal is spatially explicit in the model, and vegetation structure, kind of a baseline, determines resource competition and disturbance. What we are showing here is the difference between if we use a seedling establishment map based on that actual seedling experiment data on the maps on the right, that the landscape looks different than if we just used habitat suitability map based on adult tree distributions, as is often done, which are the maps on the left. And the T1 and T2 are two different time periods. And what we found is that those simulating the forest dynamics based on the species population dynamics based on the adult distribution or the adult suitability instead of seedling recruitment overestimated blue oak abundance. So the box plots represent the abundance, a relative abundance at the end of the simulation if we use the adult maps versus the seedling maps. And that difference was greater than even the temporal window, which is what the two different similarly colored box plots are representing. Okay. So what you have, so that was my third example. And what you will have probably noticed is, especially in the last couple examples, was a lot of terrain patterning. So biophysical process models characterize the environment experienced by organisms as a function of this scaling interaction of climate and land. This is an idea that has been promoted for a long time, for example, by Mike Austin. And so I want to show examples of a couple of different biophysical, so Austin basically argued that for this, environmental niche modeling or habitat suitability modeling, we can't just rely on macro scale climate variables alone. So I want to show examples of a couple of different of these biophysical models that we've relied on heavily in our work. So the first example is the basin characterization model, which is a water balance model that simulates interactions of rainfall and temperature with topography, soils, and geology. So for example, the effect of terrain on solar radiation is calculated because it affects potential evapotranspiration. So this water balance model and the effects of soil and geology on recharge and runoff are incorporated. So this is a grid-based model. It's at an amazing 270-meter resolution for California, and annual, seasonal, and monthly values are available for both historic climate and future climate projections. And again, you can see this, if you look carefully, the climatic water deficit, this difference between potential and actual evapotranspiration is estimated by the model, and the authors, Laurie and Alan Flint and others, the developers of the model in their paper showed, for example, two different little landscapes in California where they're showing the difference between current and future climatic water deficit with increases in deficit in red colors. And what's again really apparent is the terrain or topographic patterning in that important variable. The second example that I'll share is these ecologically relevant maps of landform and physiographic diversity developed by Dave Theobald and colleagues for the entire conterminous US at very high resolution, mapping this hill slope sequence, hill slope classes that, as physical geographers, we know are strongly related to, soil depth, soil development, and wind exposure, subsurface water flow, and all kinds of other things, and therefore also to biotic distributions. We can then see that that those interactions of terrain and climate manifest as topoclimate is a strong driver of species distributions at the macro scale. Again, showing an example from our work, endemic pine to California floristic province, Pinus sabiniana, with the changes in climatic water depth from current to future climate, showing where drying is likely to occur and where it isn't on the left, and the corresponding patterning within the range of this large ranging species with decreases in habitat suitability in the darker colors on the right side of the distribution, and in the Sierra foothills, and less increasing suitability in the sort of the central coast. All right, so I want to acknowledge my funding sources and National Science Foundation and California Strategic Growth Council, and also data sharers and collaborating agencies, and just conclude by saying there's lots of exciting developments linking genes, populations, ecosystems, geophysical landscapes, and climate over larger spatial scales. And I haven't even talked about remote sensing, but knowing who's on the panel today, I'm really hopeful that somebody else will. Thank you for your attention.
Thank you so much, Jen, for this wonderful presentation. Very informative, very important insights presented in the talk. And we are going to open up for questions. Before I ask others ask a question, I will ask you the first one as a chair. And so this integration of climate factor and the land use change is hugely important to predict what would happen on biodiversity in the future. My question to you is, for land use change, a lot of how did you model the land use change in your model? Especially a lot of land use changes are due to the factors not just locally, but also far away, sometimes thousands of miles away from the location you started in. So yeah, I
Yes.
appreciate any insight. Yeah.
Yeah, that's a really good question, and that's a really good point. And maybe the only thing that is notoriously more difficult to predict than the specifics of specific patterns of climate change is our patterns of land use change. And so, because that just involves not just the physics and chemistry of the atmosphere, but of course, what people will do. In the example I showed, I actually used published projections from the integrated climate land use scenarios. So those are based on human demographic growth models that are consistent with the IPCC special report on emission scenarios. So they're actually driven by global climate change scenarios. And they are, this was, I think the particular projections we're using were produced nationally. So they're not global, but they're national, and they are, I guess, taking some of those global factors into account, at least through climate change. But we have also used other approaches in our work that were more, we've because so much climate land use change in California is in the form of urban growth, that we've used urban growth model simulation models in other aspects of our work, but those don't necessarily explicitly account for climate change given factors.
Yeah, thank you. Yeah, that's very helpful. Ask you another question. You mentioned the statistical learning methods in your model. Now, given the rapid development in AI, what kind of future you see the function of AI in the future forecasting model?
Let's see. I think I think that some of us have been using models for a long time that we would call AI. We just call them machine learning models, and those include, things like artificial neural networks and random forests and things like that. And I just methodologically see those being on a continuum, a gradient of statistical to AI or machine learning methods, and so which is why I use the term statistical learning, which I borrowed from the textbook by Trevor Hastie and others. But I think that AI is amazing, especially when I see my students and postdocs use it, for example, for image classification, where we've done things like identify tree species from hyperspectral information. And on the other hand, I don't think AI is necessarily AI is really good at learning patterns, but I don't think it's so great at forecasting the future. Because you're just extrapolating beyond the training conditions. So I don't think that it's going to necessarily overcome that issue.
Great, thank you. I saw Stephanie and then Ines.
Thanks, Janet, that was a very thought-provoking talk. And then your comments just now are very thought-provoking as well. I'm torn between writing down some of the things you just said and asking you my question. So, my question is about sort of our tension we have in using averages when we do scaling. Because this has come up in talking with some of my colleagues about the use of averages as we do spatial scaling and how inappropriate that can be. And I think when you were talking about the seedling establishment being this critical window, clearly averages would be inappropriate in many ways for doing that scaling. But I wonder, rather than sort of plunging into a feeling of hopelessness about how many critical stages we need to know for the many different species that we care about in these biomes, did you see some ways to generalize what you learned about seedling establishment, maybe critical stages for species as we try to scale up across time and space, but also across multiple species and communities?
Yeah, that's a good question. This is a very plant-centered perspective, and so somebody else might want to chime in who thinks more about animals, but I would just learn from my colleagues who are the population modelers among my collaborators, who, when they talk about modeling population dynamics, they talk about age or stage-based models. And the whole idea of a stage-based model is that there's a sort of discrete number of steps within which you can comfortably average demographic rates, vital rates. So why not sort of learn from the population biology that we know? And in terms of scaling up to multiple species, again, maybe there are functional types that we can, that we can make some generalizations instead of having, based on sort of demographic and other functional traits that would allow us to generalize across species. So for those trees, that very long-lived trees I was talking about, I sort of think it's a pretty discrete number. It's like seedlings are very different, than adult trees, but you don't need a whole lot more subtlety than that. And yeah, I'm not sure I answered your whole question, but
I think that was great. I think you're pointing to some other areas where we can learn things that some of our subdisciplines have already worked out. I think that's great.
I mean, the other interesting kind of direction that people have gone in that area is, instead of modeling the distributions of species or of genes, like we saw in the first example, they've modeled the association between environment and the demographic rates themselves. So, integral projection models and so forth, which I think is really cool work as well. But you need the data.
Yeah, data are really important. Thank you. Ines.
Hi, Janet.
Hi, Inez.
Yeah, great talk. So looking at your work, it seems that you are pointing out that gradual transitions based on how these demographic stages may be affected by changes in environmental context. I wonder if you have ever encountered a scenario where you see an abrupt change, a complete turnover of an ecosystem, and if you did, what drove it, and if you have model it in several places or systems, was it always driven by the same driver or stage, or is this also context specific?
That's a good question. So you're looking for like a tipping point or a novel ecosystem or, I mean, there are certain, sticking with the sort of species focused work that my collaborators and I have worked on, we certainly see under some scenarios, abrupt or precipitous decline in a particular species. And so so for example, with plants in California that are in this Mediterranean climate system that are adapted to fire-prone ecosystems, a number of them have life history traits that they're basically adapted to a particular disturbance regime. For example, they may have seeds that only germinate following fire, but the adults have to live long enough to build up a seed bank in order to reproduce. So those so-called obligate seeder species require fire for reproduction, but it can't be too frequent or they never mature. So under scenarios where fire frequency gets very high, under scenarios of some combination of land use change and climate change, which both affect fire frequency in this landscape, maybe land use change more than climate change, because fire in the southern part of the state is very ignition driven, which is human driven. So anyway, that's an example of under some scenarios, you just, you have basically population extinction. So there, I mean, one other example that I can think of is, and I don't know how, what's abrupt to an ecologist, right?
So I have a kind of follow-up question. So for example, I think it was black oak, that was the name. If black oak disappears because it's not able to recruit, does it mean the whole ecosystem will turn around into something new, like a shrubland, for example? Or
yeah, no, that's a good question. Okay, so not necessarily based on our work, but based on some interesting actual observations of changes in forest structure in California in the last 60 or 70 years. Jim Thorne and others at UC Davis have shown by, basically using forest inventory data and comparing contemporary data to data that was collected, say in the 1930s, there has been this sort of large scale shift in the lower montane forests from being more pine dominated to being more oak dominated, and also to being much more dominated by small trees. So sort of losing the old growth. And so, also the recent drought in California had led to a lot of tree death that was concentrated in certain species. So ponderosa pine, for example, had a lot of tree mortality. So at what point do you come back to a landscape and it's, do we call this an oak woodland now instead of a pine forest? And how many, how gradual or abrupt does that shift have to be for us to notice it?
Great, thank you. Any other question from the committee or other speakers? All right, I will read a question from the audience. This is from Jinfeng Xiao. He said, Dr. Franklin, I really enjoy your talk. Can you share your thoughts on about the relationship between two branches of ecology, landscape ecology and global change ecology?
All right. I think that I think that global change biology, has I think that landscape ecology is one subdiscipline that informs a global change biology. And I think what it contributes, besides the data and methods and traditions of working at somewhat larger spatial scales than this, the so-called 1 square meter ecological plot that I'm not sure ever existed, but I think landscape ecology also contributes these kind of foundational concepts of that landscape patterning and connections across landscapes are important drivers of ecological processes of transfer of matter and energy and movement of species and so forth. So, I guess to me, landscape is that middle scale between organisms and smaller scale processes and the macro scale, whatever we're going to call that.
Thank you. That's a very authoritative answer from a former president of the North American Regional Association of the International Association for Landscape Ecology. So thank you. And so Louis, hands up.
Yes, thank you, Janet. That was really terrific. And so I'm a microbiologist. And so I wonder what your thoughts are about, so you're primarily working on plants, but these ecosystems have many different organisms. What do you think the challenges are in terms of integrating data to obtain a more sort of holistic view to what's happening at these sites? Do you have any thoughts about that?
Yeah, I would. Let's see. In other lives, in other in other work that we've done, yeah, we're very we're very aware that sort of no matter what approach we take, you're sort of making one slice through the problem, and there are certainly other tools for approaching the problem, encompassing a different set of processes, maybe at different scales. So, for example, I'm interested in these interactions of climate, fire, land use on biodiversity. But honestly, nothing I showed you today was directly was incorporating the effect of climate on fire, which is a notoriously difficult problem, or the feedbacks. If the ecosystem, like the plant community, changes, there's actually going to be feedbacks to the to the energy balance and so forth. And so there are other approaches like dynamic global vegetation models, which would more a process is more of a process model that is directly going to account for the effects of vegetation on fire, on biogeochemical cycling, on the atmosphere, and on the climate and so forth. But they operate at a different scale, and they're addressing basically very broad functional types. They're sort of a coarse categorical as well as usually spatial resolution, but they are addressing those ecosystem processes more directly in terms of biogeochemical cycling. So yeah, I think there are, and there are also, in my group, we've worked with other types of models that more directly simulate the dynamics of, say, the whole plant community, not just one species. And those models, such as the one's called the Landis model. It's a forest succession and disturbance model, also has sort of ecosystem models as well these days that will address carbon dynamics and so forth. So, all models are wrong, but some are useful, right? But there are some tools in the toolbox for addressing these different components of scale and process.
Great, thank you.
I have a question.
Yeah.
I was really interested in seeing the genetic phylogeographic turnover studies you showed.
Oh, but don't ask me any questions about it because I won't be able to answer them.
Okay, well, I'll just pose the question.
Okay.
See if you have thoughts. So I'm just wondering about how you see the relative importance or integration of like controlled experiments to look at response to changing environments versus like reciprocal transplant experiments versus functional genomic approaches, like what you showed, I think what you showed. Just curious how you see those different kinds of approaches.
Yeah, maybe you should answer that question because I'm I'm not sure I can. I know that I've read enough about it to write a decent proposal, but I don't do the genetic work myself, and I would always be working with collaborators. So if I understand the question, because I work a lot with trees and so forth, I know that we do have this rich legacy of reciprocal transplant experiments that have taken place, much of them in the context of commercial forestry, right? And so forth. So if we're talking about non-food crop species, it's probably one of the best. There's a rich resource of information there, which then whereas the example that I'm familiar with Arabidopsis, that it's really exciting work. And so maybe that's an example of that functional genomic approach, and they've related drought tolerance in Arabidopsis thaliana to they've related the genetic underpinnings of drought tolerance to its the distribution of that genome across Europe. But yeah, I don't. So I suppose we'll just keep knowing more about more species besides Arabidopsis. I mean, there's great folks in California working on some of the oaks in forest experiment stations, as well as their laboratories, like Victoria Sork. So I hope that sort of functional genomic approach is the way of the future.
All right, thank you. I have another question from the audience, from Xiao Jianmin and Xiao, and he said, "Thank you for your great talk. You mentioned about the small scale to larger scale processes and pattern. Can you share your thoughts about the effects of disturbance events, such as fire, ice storm, on the upscaling process and modeling?"
Well, let's see. Yeah, the disturbance disturbance regimes are something that I've always been interested in, and their influence on landscape patterns. The examples that I showed did incorporate those effects, although it wasn't obvious in my presentation. They can be one of the criticisms of the niche modeling approach that has been leveled at the niche modeling approach is that it doesn't incorporate disturbance. doesn't incorporate a lot of things. It mainly is based on environment species distributions, but it can be incorporated in kind of the more population or community ecosystem simulation modeling, and we did incorporate it into population models, such as the BioMove example, as a disturbance regime with a particular characteristic, size, frequency, and so forth on the landscape. So it's a very important factor in patterning population dynamics, certainly for plant species and disturbance-prone ecosystems, the area that I work in.
Yes, thank you so much, Janet, for your excellent presentation. I wish we have more time. Maybe we can get back to you for more insights from you in the future. But we have to end this portion of the session today here. But now I would like to invite Louis, one of our wonderful committee members, to moderate the first panel. Louis, please take away.
Okay. My name is Louise Glass. I'm at the University of California at Berkeley in the Plant and Microbial Biology Department. And today we have 6 panelists that will be presenting their research on different scales of biology. And first of all, I'd like to thank all of the panelists for their time and effort in being here and their presentations and answering questions from both the committee and the outside audience. The goals of this discussion are to explore the ways that small scale biological research informs larger scale research efforts, and how those larger scale data actually informs directions of small scale biological research. And importantly, how the specific knowledge and methodology and approaches and tools can be translated across these multiple scales of biology. So the panelists will all have about 10 minutes total for their presentations, about 8 minutes of talk and 2 minutes of question and answer. But at the end of the panel, we will have 25 minutes for question and answer session, so that we will hopefully have time to address all of your questions, even though we might not get to them right after the talk. So it's my pleasure to begin introducing the members of the panel. Are we ready to go? Okay, great. The first speaker is David Schimmel. He is a senior research scientist at NASA's Jet Propulsion Laboratory, and he will be discussing gaps and biases in our knowledge of global ecology and the role of remote sensing in both revealing those gaps and aiding in the extrapolation and generalization of concepts. David?
Thanks very much. I'm going to be presenting just a few examples of what is really a revolution in our ability to quantify some of the processes, the patterns, and aspects of the diversity of life itself from space. And I'll be presenting work that really represents activities by a huge community that has brought a number of new measurement capabilities to the table over the past decade or so. Next slide, please. I just want to remind us of the blue marble. This image is based on 2 long remote sensing time series, what we call greenness on land, the absorption of photosynthetic light, photosynthetically active light, and chlorophyll in the oceans, giving kind of the signature of the large scale continental to global scale patterns that scale with climate, with orography, with phylogenetics, and that we use to understand the Earth as a system. The next slide, please, really shows what we have to worry about as well, the heavy footprint of humanity. This is an image from a paper written by colleagues of mine showing some of that impact in terms of urbanization, transportation, land use change, affecting really all of the ecosystems of the Earth today. Next slide, please. I'm going to really focus on an old paradigm, but it's a good one in ecology that we do our best when we're considering several scales at once. And in this case, going between a sort of a process-oriented approach based on ecosystems and organisms, and both informing and evaluating the generalization of that underlying biology with global scale observations. Next slide, please. I began doing continental scale analysis a long time ago, really at a time when we only had models and very, very limited ability to gather data at large scales. The satellite vegetation index and maps of climate and soils that were based really on a very limited number of points. Next slide, please. Whoa, not sure what happened here. That was completely not what is on my version of the slide. Can I switch to sharing myself? See if this works better. Can you see my slide? I hate saying this. Can you see my screen now?
Not yet.
Well, let's go back to the other display. That's not working. So what this slide is supposed to show is a plot of carbon storage and productivity of the biosphere, the land biosphere, scaled across latitude from the South Pole to the North Pole. And what it shows, as you might imagine, is most of the photosynthetic activity and much of the carbon storage occurs in the tropics, with high carbon storage also in the boreal and at high latitudes. But when we look at the experimental and observational data that we use to understand continental to global scale ecology, it's biased away from those two most productive, most carbon rich, and most biologically diverse parts of the world. In fact, most of the data at the 70 to 85% level that we have for species, for genes, for physiology has been collected in the mid-latitudes. And when we try and upscale that information, we wind up with artifacts that are very difficult to remove from our analyses. Next slide, please. So this is an example of the power of remote sensing to change our view of the world. We've known for many years that during El Niños, the biosphere releases carbon to the atmosphere. We see increases in the growth rate of CO2 in the atmosphere. And we know for a variety of reasons that change is due to dynamics in the tropics. But we've always, because we had just global carbon dioxide data and very simple biome models, we always assumed tacitly that the tropics behaved as a unit. But one of the very first results from our orbiting carbon observatory, which combines a measurement of CO2 with a very direct correlate of photosynthesis, solar-induced fluorescence, and takes advantage of carbon monoxide as a wildfire tracer, showed that each of the 3 tropical regions responded to El Nino for different reasons, with Asia being dominated by fire, Africa being dominated by an increase in respiration, and really only Amazonia behaving the way we had imagined for decades, with a drought-related reduction in photosynthesis. So when we begin to see more and more of the world with sampling that isn't biased to where we live, where our wealthier institutions are, where our wealthier funding agencies are, we're learning new things about the world that are continually surprising us. Next slide, please. This sampling bias is equally true for biological diversity. This shows plots from one of Janine Cavender-Barr's recent papers with points that have been sampled for plant functional traits, a key diagnostic of how plants behave, overlaid on diversity hotspots, and a measure called contextual intactness that deals with the extent to which direct human land use has fragmented the landscape. And what we see is a systematic, again, a systematic bias away from some of the most diverse and most biologically important parts of the world. Next slide, please. We're beginning to have remote sensing tools that can really begin to reveal these patterns, though. So the image that you see here is going back to some of Janet's work, is the California coast, the Mediterranean ecosystem, Point Conception, sort of where California goes from going east to west to north to south, a region that goes from coastal ecosystems through chaparral, oak woodland, and eventually high altitude coniferous forests. And on the right, we see seasonal dynamics of a mosaic, the step beyond remote sensing of greenness, showing an RGB map of 3 plant functional traits: chlorophyll, leaf water content, and leaf nitrogen, over the course of the phenological progression of the year. And so we begin to see, first of all, patterns of functional diversity and patterns of temporal diversity as this extraordinarily complex and biologically diverse landscape changes over the season, where we see both the developmental biology of individual species, the leaf out, for example, of oaks, but we also see the replacement of early season species by mid-season species, by late summer species, dynamics that are hinted at but not revealed by our long greenness time series. Next slide, please. So we now live in a world where we can make many of the same measurements at plot scale or eddy covariance site scale, where we measure carbon dioxide flux, radiation, leaf area, forest structure. We often sample foliage to make measurements of plant functional traits. And of course, we measure biophysical properties. From space, we can now make many of those same measurements using emitted and reflected radiation, as well as active remote sensing radar and LIDAR to retrieve forest structure. And the scales of those remote sensing measurements vary quite a bit, in the same way as the local scales of our in situ biology measurements vary from, for example, the height of an individual tree, to the density of stomates on an individual leaf, to the diversity of the genome in a small sample of soil. We can measure carbon dioxide flux at 100 or 300 kilometers. We can measure solar-induced fluorescence at 1 or 2 kilometers. And we can measure canopy height at, let's say, 15 to 30 meters. So we begin to be able to put together these different scales of observations providing both a top-down constraint and questions about change over time that really have to be answered by going back to the more organismal scale. And so I think we're entering in a kind of a golden age of continental to global scale remote sensing, where things that were only accessible to us on the ground with biased and sparse sampling as a necessary consequence, we can now work synergistically with airborne, drone-based, and space-based remote sensing to combine scales and understand biology in an integrated way across a wide range of scales. Thank you all.
Thank you, David. That was great. I think we have time for one question. Martine, I saw your hand up first. Oh, it's not? You're clapping. Oh, okay, sorry.
Well, I'll take that too.
Anybody have a question for David going forward? We can also address it at the final question period. Okay, thank you, David. I actually have a question, but I think I'm going to hold on to it until the end because I think it's might be a long question. Is that okay?
Long question or a long answer?
Yeah, maybe both. Yeah. Okay. All right. Thank you very much. I really appreciate it. It's very interesting. Okay, our next speaker is Emily Elo-Fadros. She is a staff scientist at Berkeley Lab, where she heads the metagenome program at the Department of Energy's Joint Genome Institute. And she also leads the DOE's multi-lab National Microbiome Data Collaborative. And Emily will be discussing how scaling computational tools, data resources, and interdisciplinary collaborations will enable access to a genomic blueprint of the Earth's microbial communities. Thank you, Emily.
Great. Well, thank you so much. It's really an honor to be here today. I've already learned so much. I have just 2 slides that I'm going to walk through today. And I think it's great to go after David, because thinking about this golden age of remote sensing, I'd argue that we're in the golden age of microbiology. And so I wanted to share a perspective from Berkeley Lab, where I work with an interdisciplinary team at the Department of Energy Joint Genome Institute. And I'll also be discussing this relatively new program that Luis mentioned, where we launched out of JGI, this National Microbiome Data Collaborative. For those of you who may not be familiar with the JGI, it's a DOE national user facility that provides genomic resources to the global scientific community. At the JGI, our team uses metagenome sequencing to explore really the diverse array of microbial communities to try to identify and characterize genomic information from uncultivated microbes and viruses. We use an approach called genomes resolved metagenomics, which can computationally reconstruct genomes from the environment without having to culture them. Over the past several years, we've developed standardized bioinformatic workflows to scale to thousands of metagenome data sets from diverse environments that include terrestrial hot springs, Arctic permafrost soils, and of course the coastal oceans, which are very important when thinking about larger scale biology. So today I wanted to share a short story about a community resource project called the Genomes from Earth's Microbiomes. And this was an effort to catalog microbial diversity. And I wanted to especially highlight how efforts like this can support a broader vision of continental scale biology. For this project, we set out to demonstrate really the value of performing genome-resolved metagenomics at scale, and importantly, how we can develop accessible tools for the research community to explore the data and to analyze these genomes beyond our immediate group. At the time, we took over 10,000 metagenomes sampled from this diverse microbial habitats and geographic locations and annotated over 52,000 composite genomes using our newly developed standardized workflows. I'm showing a snapshot on the left-hand side here that shows the breadth of terrestrial samples, where the size of the circle represents the samples that were used in the study. But as you'll note, and what David mentioned, there are these huge sampling biases that you can very readily see here in this map, where areas like South America and Africa are just not represented at all. And so this highlights the need to continue to broadly sample diverse biomes across all continents. Next, we clustered these genomes and identified about 18,000 species level operational taxonomic units for those microbiologists. This is a pretty rough gauge of species classifications that I won't get into, but this is a typical measure of describing microbial species. And these species level groups really captured a broad phylogenetic and functional diversity of uncultivated bacteria and archaea. And we used this catalog to compare at the time to about 500,000 reference genomes and found that there were about 12,000 likely new species, of course, all computationally predicted. When we place these genomes using concatenated gene markers shown in the middle panel here, we were able to demonstrate that the overall diversity was really immensely expanded. All of the green coloring on the inside phylogenetic tree were new groups that were added by this data set. And so it increased the expanded archaea and bacterial diversity by about 44%. From this increase, we were also interested in generating metabolic models for these genomes. So trying to look at and predict the biogeochemical functions that could be encoded within these genomes, because as we know, microbes are the microbial engines that drives Earth's biogeochemical cycles. And so this was done in collaboration with DOE's systems biology knowledge base. And so in addition to just expanding the known diversity of microbes from this sampling, we were really interested in looking at biosynthetic gene clusters and using this data to resolve viral host linkages. I'd also like to mention that in the spirit of building scientific community and recognizing the global contributions of all of the research teams and researchers who went out into the field to do sampling, we developed a new consortium-style authorship model where we included over 200 researchers as part of this community resource. The GEM catalog, while we were really excited that it expanded the known microbial diversity, it was really surprising when we went to map back the reads from all of these metagenomes to the catalog that we generated, and found that there were still 70% of reads that were only mapped to a variety of environmental samples like soils. And so I would really highlight that we just scratched the surface using this type of genome resolved metagenomics, and there's still a lot of work that can be done in this area. So since the GEM catalog came out as a community resource in 2020, our team has continued to develop and expand this workflow to scale to even more data, to integrate additional updates in taxonomic and functional annotations, and then importantly, to incorporate eukaryotic genome identification to expand beyond just bacteria and archaea. We add on a monthly basis about 300 new 3,000 new genomes as part of JGI's production scale workflows. And so if you go into IMG, which is our platform for analyzing genomic and metagenomic data, there's almost 230,000 composite genomes or metagenome bins from over 21,000 metagenomes, which can be interactively explored and analyzed within IMG. I also wanted to mention that we've increasingly been using a new tool for parallelized metagenome assembly, which is called MetaHibmer. This tool was developed by Kathy Yellick's team for the Exabiome program at Berkeley Lab and can assemble terabase scale data and generate genomes from even the low abundant or rare microbes within a sample. So we can computationally combine metagenomes from either similar samples or similar experiments and leverage MetaHibmer and differential coverage abundance patterns for metagenome binning to recover even better representation of the microbes in any given sample. And so this scale and resolution, I believe, has the potential to genomically catalog much more of the microbial diversity on Earth and start to chip away at that 70% of unmapped reads. All right, next slide, please. I also wanted to briefly discuss complementary work that's being undertaken at the National Microbiome Data Collaborative, where we're addressing some of these broader challenges in microbial research. So for microbial ecology, I think many of you can appreciate that data generation has really outpaced our existing infrastructure for how we collect and process and distribute data in a uniform and accessible manner. And so the NMDC was really developed to integrate data that spans not just sequence data, but also proteomes and their metabolic footprints. And so we can integrate all of this information along with the contextual information like latitude, pH, or when and how a sample was collected. The NMDC sits within this larger ecosystem of data resources and is heavily focused on supporting data sharing and interoperability across the existing infrastructures like the JGI. The NMDC focuses on data standards, infrastructure, and interdisciplinary community building across the environmental sciences and is developing resources to lower barriers for data submission and reuse. I'd also just like to highlight that the NMDC has been working very closely with the Genomic Standards Consortium to translate reporting standards that have been in existence for well over 15 years into machine actionable standards that will eventually lead towards machine learning for microbiome data. And so lastly, I wanted to highlight the concept of national user facilities and national resources like the JGI and NMDC that have really been important in supporting research through large-scale team science. Having the sequencing power, high-performance computing, and genomics experts has really enabled the cataloging of microbial diversity at the JGI. And similarly, investments in data standards and ontologies and technical implementation of those standards like the NMDC is necessary for maintaining and innovating to keep pace with the science. So thank you again for the opportunity to join today, and I look forward to the discussion and happy to take any questions.
Thanks, Emily. That was great. So we're a tad bit behind. And so the suggestion was made by the higher ups to hold all the questions till the end. So thank you again. It was a great talk. And I have a couple questions that I want to ask you in the Q&A. Thank you very much. Okay. The next speaker is Dr. Sydney Record. She is an associate professor of landscape conservation in the Department of Wildlife, Fisheries, and Conservation Biology at the University of Maine. And Sydney will discuss how synergizing across environmental observation networks has the potential to transform continental scale biology.
Let's see, should we put it? Okay. Thanks so much for having me here today. Next slide, please. So we're at a really exciting time in the history of continental scale biology. We've got data that span across space. NEON was built out in 2019. We also have long, lengthy time series. So LTER just turned 40 recently. And we have data that also span scales of biological organization, from genomes to whole organisms, all the way up to ecosystems. Next slide, please. But I would argue that we're not going to fully harness the potential of these different data streams unless we can make connections across these networks of data collectors and providers. And this is very US focused, but I acknowledge that the challenges that Dave mentioned in his talks about global analyses. So today I'm going to talk about a case study from my own research where I found synergies from the LTER and NEON networks that really have helped to inform the continental scale analyses that I do in my own work. So next slide, please. So the motivating question in this research was what influences biodiversity from regional or continental to local spatial scales. So generally, when we're trying to understand how communities assemble in a local community, we consider that kind of on the top part of this graph, we have a regional pool of species. These are shown by different colored curves here along a trait axis. So there's all these potential species with different traits that could end up in an area. But there's external filters, such as large-scale climatic variation. Maybe, there's species may have physiological intolerance to a particular location. And so at any given locale, we end up with a subset of those species. They can then experience that subset of species in internal filters such as biotic interactions, microhabitat heterogeneity, and then kind of partition their niches along that trait axis even more. And that's the observed biodiversity that we see. So we're oftentimes trying to understand what are these drivers of biodiversity at these different scales. Next slide, please. So with that motivating question in mind, my colleagues and I started working with early NEON data. And we wanted to explore drivers of small mammal diversity with that data. And the NEON data is really incredible for these kinds of questions because they're community biodiversity level questions, but they're really informed by information on both intra and interspecific variation in traits. And with the small mammals, NEON collects individual size data for every single small mammal that they trap across these communities, across sites, across the continent. And with that data, you can build those size distributions that I showed you in that last graph. And these are examples from 4 of the representative NEON sites. And you might look at things like the amount of overlap in those body size distributions, which could give you a sense of biotic interactions in terms of the potential for competition for species of similar body sizes for resources. So these graphs are arranged from top to bottom from kind of high amounts of overlap or potential competition to lower amounts of overlap. And those actually, in this case, happen to coincide with a temperature gradient from cold to warm. As we're looking at that data, next slide, please. We were, thinking about, okay, so you can look at how this variation in overlap in body size distributions differs and, maybe with large scale climatic variables. But we were wondering, there's quite a bit of variation on that y-axis in overlap. And what might that be due to other than just temperature, right? And these stars show some of the individual sites that are plotted on that graph. And in particular, since I've done a lot of work with the LTER network, I was very curious about what was going on at these different sites, right? So as an end user of NEON data, you kind of are trying to do this analysis across this huge continental scale, but it's a bit weird because you haven't been to all of the sites and actually experienced all of the conditions that the organisms are experiencing. And this got me to thinking that, next slide, please. At NEON sites, we have them measuring many aspects of the ecological systems, all the way from the small mammals and the beetles scurrying around on the ground, up to the amount of CO2 that's in the atmosphere above the sites with the eddy flux towers. Next slide, please. But we're really missing some key information still, right? To understand the response, the variation in our response variables that we see, such as disturbance and land use. And part of why these variables are missing pieces of the puzzle with NEON is because they were descoped during build out of the observatory. So I know because I've done work with the LTER network at Konza Prairie, for instance, which was that site in Kansas, that since this is a co-located and LTER network site, I know from the LTER network that there's actually a long-term experimental grazing and burn study at that site. And if you go and you plot all of the small mammal trap locations, you can see that they span that gradient across those different manipulative experimental prescriptions, right? And so this gave me pause in terms of working with NEON data without this site level context, right? Because at a continental scale analysis, you might attribute variation in your response variables to climate, but indeed, if you don't include the land use, land management, and disturbance in your model, you're going to maybe have erroneous inference. And so I then began for the last 6 years, I've been developing a past land use database for NEON sites and a current land use database. So if you go to the next slide, please. This shows you some of for the forested sites in NEON, some of the natural and human-made disturbances that these sites experience. So cutting, both natural wildfires, prescribed burns, planting for regeneration. Next slide, please. And at some of the, next slide, please. At some of the rangeland sites, you can see that there's grazing, there's also natural and prescribed fire, even some chemical treatments, right? So next slide. What I would argue is that looking ahead for all of these different networks, we need to know not just kind of where they are in a climate envelope in space, but also the land use and management disturbance context of the sites, right? Because at worst, we might kind of have erroneous inference about where we're attributing the variation in our data. But maybe more importantly, right, unless we have that kind of human context in our analyses, we won't have science that's going to be relevant for managers and society and my kids, right? Next slide, please. So this is just one example of how we've I've taken synergies across a network of networks to inform the kind of continental scale analysis and the inferences that we can make across a continent. But I would argue even further that all future innovations in continental scale biology will potentially come from this network of networks approach. And in this ecosphere paper that we recently authored, we talk about how connecting people, data and tools and infrastructure across this network of network landscape is going to be really integral to making sure that we're able to innovate in the types of inferences we can make at large spatial scales and across time series and across scales of biological organization. And next slide, please. So thank you. I look forward to your questions in the end, and all my references are on the next slide for the people who are on the committee. Thank you very much.
Thank you very much, Sydney. That was great. All right. So the next speaker is Martin Winter from the German Center for Integrative Biodiversity at the University of Leipzig. So Martin will be discussing limits to data-driven synthesis with a focus on global biodiversity and framework needs, including monitoring to track biodiversity change, biological invasions, and species traits. Okay, thank you. Take it away.
So, hello, can you hear me? I guess so, right? Yes, I'll have to talk quickly, and I don't show so many pictures, but a lot of text. Okay, let's start. What is it from my perspective? I mean, it's my own perspective as researcher, as someone who's seen a lot of research. What limits to continental and global research do I see? First of all, I see a lot of knowledge out there, but very little exchange or easy findable information, which leads very often to a huge amount of redundant work. For example, when people developing tools, so I'm a macroecologist, we use a lot of R and all these functions and tools. Many of my co-speakers here do the same. And there's a lot of redundant development of tools and varying quality as well. And one example we recently had to do, because we actually wanted to do something else, but then we had to go back and developing some overview about taxonomic name harmonization. So when you match different data sets, that's what macroecologists do, and you need a unique identifier, usually a species name. And there's a wild, wild west of things how to do that. And what we did, we developed, not only we wrote a paper about a little guideline, let's say, from different perspectives, but we also developed a little shiny app. And what you can see here are triangles. So again, it's about taxonomic data. So the triangles are the databases, so of taxonomic information. The circles are packages, R packages in this case. That's the language we are working at, or the environment. And you see the dependencies. So if there is an R package using a certain database, so for example, the triangle here in the middle, it's the plant list and outdated global backbone for plant taxonomic information. And you see that there's a lot of dependencies and interdependencies between different databases with taxonomic information, like for example, GBIF as well, and tools. So there's a lot of redundant work happening because many of those tools do basically the same stuff. Or some of these tools rely on data which are not updated anymore, but still work with these outdated data, like TPL, cited, I think, 1,500 times every year, but since 10 years not updated anymore. It's just that communities not really, not always, and sometimes not communicate to each other well enough. And that also hinders some transport of knowledge from small to large scales. Moreover, that's just one example. Let me move here these panels. That's just one example, and I see way more interoperable, so problems with interoperability between data across taxa systems or research cultures, which also includes different languages. Yeah, for example, and now I'm talking not only to Brian here, that we have, and that's research I'm doing, so plants, plant traits. So we have different regional, continental, and even global plant trait databases like FRI or BN or Austrates for Australia. And they are not easy to combine if you want to work at larger scales and with invasions. Obviously, let's say Australian plants occur also in other places of the world, and you want to use the trait data. There's many things to say about that, but for example, we lack a coherent trait definitions. All these databases, for good reasons, and I don't blame anyone, came up with partially different trait definitions, which make it really hard to combine these data. If you want to work at larger scales, you have to. And another issue with plant traits, and I'm not talking about only plant traits, it's also about many other trait databases. They miss geolocation of the trait measures, and if you link species traits with other measures from different regions and environments, so much better, much worse than, for example, the speaker before about the neon data, then you can't do much about that. So you combine sometimes trait data to a species occurring, let's say in Germany, although the trait data was measured somewhere else. Yeah, that doesn't make much sense, but it is the way it is at the moment. So it results in a lot of uncertainty, especially at the user side. That's what I'm seeing a lot. So for example, which product or tool is now the best to be used? It's a diversity of different products, resulting basically in the same question. And it also leads sometimes to difficulties if you want to compare research results. The next one is again about traits, and it's not about the tools around that. It's rather about the lack of data. And this is a paper we are working on just now on my table. That means it will rest a little bit more. So we have on the x-axis the number of traits. So that is combined across several global trait databases or regional ones. So we combined them, and the y-axis is the number of species. So but with focus on alien plants. So what you see here is, let's say, the data density plot for all global alien plant species and combined 4 major plant trait databases. And what it actually shows, without that you understand the detail, is that majority of those, the majority of the matrix is empty. And the same, if you put it on a map, I'm a macroecologist, that's what we do. It's a proportion of alien species with trait combinations of the leaf height seed mass, one way to describe major axis of plant ecology, and the colors indicate the richness of the data for that region, alien plants. Just remember, it's not all plants. And you can see some regions, and especially those regions with fewer aliens, maybe also the knowledge about the aliens, might have a lot of data, almost all plants covered with leaf height seed data. But if you go in more interesting and species-rich regions, and even not only species-rich regions, you see we lack a lot of trait data for plants, in this case alien plants. So when working with communities and in being interested in meaningful functional traits and not just what is there, especially if you want to combine those trait data with local community, species community data, we see a lot of relevant data missing, and that's only for plants. Yeah, think about other important groups for ecosystems like invertebrates, much worse. Yeah, that's what I said. So at the species level, we lack comprehensive availability for data for larger spatial scales like continental or global scales. What solutions could we have? Imputations, yeah. What can you impute beyond traits? How about intraspecific variability? AI was mentioned today. It might help, and everyone is working on that, but not yet massively seeing that in the data. Mobilizing existing data, that's also an issue. Of course, we always should measure more in situ. Who funds that? Is that enough? And of course, also mobilizing knowledge from non-English literature, especially gray literature. Much of that is still printed and not yet digitally available, etc. So there's a lot of things to be done. At the moment, we don't see the results of this work, which happens, maybe not. And maybe some less sciencey thoughts on that. It's not always about science, and I leave the science to my co-speakers. What is also hindering that, and that's what I'm seeing, I'm running a little funding agency. Funds are often not funding or long enough technical data scientist, database manager positions individually. I'm not sure how it's for the NSF. I'm from Germany. That's definitely the case here. And often these projects, and even if you have these people, they're not a long enough hire to ensure data maintenance and proper database development, interoperability, blah, 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 with other structures, because their time is not enough. Yeah. And there's very little funding to build data infrastructures on a long-term funding plan. There are examples, good examples, NEON may be one, but it's not always the case. So I'm living in the center Europe, and I've been working on European projects, and there are a lot of these heavily funded databases which are now dead because nobody takes care of them, because nobody's funded to do that. And on the other side, we see also a lot of students coming in, they have absolutely zero ideas about data management. And that's on the shoulders of all of us, I would say. And another word on the non-sciencey aspects, a 2 years postdoc is just impossible to ask a 2-year postdoc to harmonize and collect data, to impute data, to write a manuscript and proposals, teaching outreach, etc., etc. And then having a good, well-built database, it's just impossible. Yeah? And that's what we ask for. And that hinders us maybe at some aspects doing great science at larger scales. And the last point I want to make is the GBF. We should have heard, all of us should have heard about that. And the new GBF targets, as also the old ones, which most of them failed, call for dynamic data interoperable, especially across scales, for example, on species invasions, but also on other characteristics like threat status or chemical flows. And that's just one example. I'm an invasion ecologist next to macroecologist. That's the wording at the moment for the target of the invasive alien species, 50% reduction of rate of introduction. But for that, we need to know the rates of introduction today and in future to track that. Yeah, that's politically super important. It will come, and many stakeholders will ask for researchers supporting developing indicators to do that. But for this need for global indicators and the need for tracking the status of biodiversity needs robust data, especially from smaller grain sizes, relatively small. I talk about countries maybe, because that's the political level the decision makers work at, and not to speak about smaller grain sizes like grids or something, and that across larger extents like continents of the globe. And I'm co-leading a project to develop one of these indicators. And what you can see on the x-axis is the evidence for any introduction of alien species. 100 means 100%. So for all known alien species, in this case, again, focusing on plants, they have the knowledge when the species was introduced. And on the y-axis, you see here, do they have evidences for impact of alien species? That's what we are actually interested in on the invasive species, not all aliens. And the size of the bubbles is for how many species do we have knowledge to model range dynamics. So certain amount of location information per a certain period of time. And what you can see here, very few places in the world have 100% of all these dimensions. And many places have very little information to build robust indicators at a country level, which is not a very specific and small scale level, to track invasions, which the GBF is asking for. And that's just one example of the many things. Yeah, so I'm talking about the lack of data in this day today. So we lack a lot of comprehensively and global available data on dynamics and impact of alien species. More on
Martin, you need to wrap up.
Yeah, I wrap up. That's invasions, and that's all. What I wanted to say was just one slide more. Sorry.
Oh, okay. Thank you. Yeah, lots of challenges there that you brought up. That was great. Okay, thank you very much. And I'm sure you'll get some questions at the end. Our next speaker is Noah Ferrer at the he is a microbial, he's a professor in the Department of Ecology and Evolutionary Biology and the director of the Center of Microbial Exploration and a fellow of the Cooperative Institute for Research in Environmental Sciences at the University of Colorado at Boulder. And Noah will be talking about how continental scale research in microbiology can be used to link microbial ecology to epidemiology with a focus on mycobacteria that live in plumbing.
Okay, can everybody hear me okay? Okay, so thanks for the invitation to participate today. So I'm a microbial ecologist, and a lot of my group's work has focused on studying small things, namely bacteria and fungi, at big scales. So looking at the distributions of microbes across space, the factors driving these distributions, and the relevance of microbial biogeography to human and ecosystem health. So today, I really have just two objectives. One, I want to demonstrate how continental scale studies, or at least US scale studies, can benefit research that integrates multiple fields, including microbiology, epidemiology, and biogeography. And I also want to show how continental scale research can be conducted using a citizen science, also known as community science, approach to benefit both the research itself and associated education and outreach efforts. And as a case study, a fairly narrow case study, I want to talk about some recent work we've been doing focusing on the genus Mycobacteria, one of my favorite groups of bacteria that includes two well-known species that you're probably familiar with, Mycobacterium tuberculosis and Mycobacterium leprae that cause tuberculosis and leprosy, indicated in red here on this phylogenetic tree. But this genus also includes more than 200 other known species, including many innocuous mycobacteria that are common in soil and water, as well as other pathogens that are indicated in blue here on this tree that can cause non-tuberculous mycobacterial disease, which is basically just mycobacterial diseases that are not tuberculosis or leprosy. And currently, pulmonary non-tuberculous mycobacterial disease affects, there's estimated to be more than 200,000 cases per year in the US alone. And many of these cases are in individuals with cystic fibrosis or other impaired immune system function. And the symptoms are, similar to what we see with tuberculosis: reduced lung capacity, persistent fever, cough, and fatigue. And it's very hard to treat. So it's, a lot of money, more than 1.5 billion and 1.7 billion per year in healthcare costs. And it seems to be increasing in the US over the last decade or so. The thing that's interesting about this disease is it's not just caused by a single bacterial species. Rather, disease is caused by multiple different species within the genus, and these are indicated in blue here. And also, this disease, unlike tuberculosis, it's not transmitted person to person. Instead, these infections are environmentally acquired. In other words, we get these pathogens from the environment. So what environment? Well, we know that mycobacteria are common in a wide range of environments, from soil to pools and hot springs to surface waters. But there's increasing evidence, and we don't really know the most important source, but there's increasing, at least anecdotal evidence, that showerheads, or premise plumbing, and showerheads in particular, are a really important source of these mycobacterial infections. They can grow inside showerheads, and when you take a shower, they're aerosolized and you breathe them in, and it can cause the disease. So this is what motivated some of this work, specifically looking at premise plumbing or showerheads as a source of mycobacterial disease. The other thing that's interesting about this disease is its prevalence is highly variable across the US. So if we look at epidemiological data, there's hotspots of disease, including hotspots in Southern California, Florida, Hawaii, and the New York City, Philadelphia area. What's unclear is why these hotspots exist. So for that reason, a few years back, we started the Showerhead Microbiome Project. And what really motivated this work is we were testing this hypothesis that these hotspots in disease prevalence are where pathogenic mycobacteria are more common in premise plumbing, specifically these household showerheads. So we recruited volunteers to participate in this study and sample their showerheads. And in addition to just using these volunteers to collect samples for us, we also involve them in all steps of the research process, keeping volunteers in the loop as the study progressed, answering questions, hosting a series of webinars, and so forth. to explain the study, the motivation, the results, and so forth. And we also use this project sort of more broadly to introduce people to microbial diversity, specifically microbial diversity found in our homes. So they sampled the biofilms inside their shower head. And I don't know if you've looked in your shower head recently, probably not, but there's lots of microbial biofilm in there. So there's no shortage of microbial biomass, despite it being a pretty challenging place to live. So they swabbed their shower heads and as well as collected some basic chemical analyses of shower water, as well as information on, for example, where their household water comes from. So we ended up with more than 600 samples from across the US. And these were about 520 municipal households on municipal water and about 86 homes on well water. And the swabs were sent back to the lab, and we used a variety of DNA approaches, including high throughput DNA sequencing and quantitative PCR to quantify the amounts and types of mycobacteria in each sample. And just a few examples of some of the results. One thing we found is that mycobacteria are far more abundant in homes on municipal water than homes on well water. And of course, there's many factors that differ between municipal water and well water. But one factor that is clearly important is chlorine concentration. So nearly all municipalities in the US add chlorine as a residual disinfectant. And we see this in the measured chlorine concentrations, which are about 10 times higher in homes receiving municipal water. Why does this matter? Because we know that mycobacteria are really resistant to chlorine. Most other bacteria that we find in premise plumbing die when they're exposed to chlorine. Mycobacteria just shrug it off. So adding chlorine as a residual disinfectant, we appear to be selecting for mycobacteria in premise plumbing. We also find lots of mycobacterial pathogens, and these are indicated with the arrows here. And this is just occupancy, so percent of samples where we found these pathogens. So unlike other potential source environments like soil, where pathogenic mycobacteria are really rare, we see lots of them in the shower head. They grow and accumulate in the shower head. You turn on your shower head, they have potential to be aerosolized, and you can breathe them in. And what's interesting is that if we look at, here's just 3 examples of 3 pathogenic mycobacterial groups, they have a biogeography. So they're not evenly distributed across the US. So for example, if we look at Mycobacterium abscessus, which is a well-known pathogen capable of causing pulmonary disease, it tends to be in higher abundance in Florida and Hawaii. And same for some of these other pathogens. They're not evenly distributed in households across the United States. So I'll spare you a lot of the statistical detail, but we work with epidemiologists at NIH to relate these patterns in mycobacterial distributions in premise plumbing, specifically shower heads, to the epidemiology of the disease, and they're surprisingly well correlated. So in other words, we can predict which area have a higher risk or higher prevalence of mycobacterial disease by looking for the distributions of these pathogenic mycobacteria in household shower heads. So where you live has a large influence on your risk of exposures to these pathogens. So hopefully I haven't made you afraid of your shower head. That's not the goal here. That's not my intention. In fact, this was one of the challenges of using a citizen science approach, is how do you communicate our findings without making people paranoid? These pathogens appear to be common in shower head, and their abundance in shower heads, and their abundance of these bacteria in your shower heads can increase your risk of acquiring the disease, but the vast majority of us are not susceptible to these pathogens. So I just want to finish by sort of, hopefully I've convinced you just with this short case study of the further convince you of the benefits of continental scale research. But I also want to highlight some of the benefits of using a citizen science-based approach for community engagement and opportunities for outreach and education. I think clearly the best way to learn about science is to participate in science. And also just on a more practical level, using a citizen science or community science-based approach makes it feasible to conduct studies across broad spatial scales. But there's an important caveat, and that is that study design and implementation of the project has to be really carefully considered for many reasons, including some obvious reasons. But I think there's a lot of potential to sort of leverage these citizen science approaches as we move forward and conduct additional studies in continental scale biology. Thank you.
Thank you, Noah. That's the first time I've ever seen a picture of Psycho in a talk. It's really great. And again, we'll take questions at the end. The last speaker today is Thomas Elmquist. He is a professor in natural resource management at Stockholm Resilience Center at the Stockholm University. And his talk will be focusing on urbanization and scale and how biodiversity and drivers of change are affected by urbanization. So thank you, Thomas.
Yeah, thank you very much. And thank you for inviting me. So this area, analyzing urbanization, biodiversity and scale, has been overlooked for quite some time. But recently there's been some interesting development, particularly based on new sets of data that's been available thanks to, among other things, of course, remote sensing data. So the next slide, please. And there are some patterns emerging. And so here is one pattern that you often find, that you have a difference just in the species area relationship between urban and non-urban areas, which have many sort of causes, of course, and factors that influence that. One, I think, very complex one is related to how different drivers interact and how they scale, and different relationship with scale in urban and non-urban areas. We could take you could click the next. I think there's yeah, so multiple drivers have different scaling. Oh, go back, sorry. Different scaling relationships in urban areas compared to non-urban. Of course, one obvious factor that is important here is that we have nearly always a high occurrence of non-native species in urban areas. Next, please. But it leads to that urban areas often show rather high alpha diversity. And in this, I wanted to illustrate through time with urbanization and globalization, we have moved from a world with lower alpha diversity, but higher beta diversity in urban areas, to a world where we today have, with time and age and also urban expansion, higher alpha diversity, but lower beta diversity, particularly if you adjust for latitudinal gradients. So it tends to be a similar sets of species occurring in urban areas, but in each specific location, they might tend to be rather species rich. Next slide, please. The other scaling patterns that are of interest, where you look at urban area and different variables and the slope. And you could have, like here, 3 examples of, you could have a beta slope that is greater than 1, equal to 1, or less than 1. Interestingly, slope greater than 1 is found when you look at green space in Europe in relation to city size. And this is based on a study by Uchida et al. Except for UK, so it would be interesting to understand what happens there. But a similar slope will also be found for other interesting variables in urban areas, like the rate of innovation also scales in the same way. Then you have more of a constant per capita scaling, like water use. And lastly, you have economies of scale. That could be public transport or built environment, for example. But the interesting observation here on green space and the slope raises questions about governance mechanisms as city grows, and whether also finance and innovation plays a role in providing more habitat in the urban area. Next slide, please. Related to that scaling discussion is also a shifting patterns of modularity and connectivity with urbanization and globalization, moving from a world with high modularity and low connectivity, although we could have highly connected local nodes through an intermediate modularity and connectivity to a world where we have a low modularity and high connectivity. And so they're highly connected globally and potentially poorly connected locally. And this could have lots of direct consequences for biodiversity, but also indirect consequences, for example, on flow of information and governance issues. Next, please. So I want to end with just a number of questions that I think are important to ask. And I think we could probably dwell on answering and discussing the rest of this session. But I think one of the most important one is try to understand how governance, the relationship between governance and scaling, and how you could understand those relationship when you also understand patterns of biodiversity. And I think there's a lot of more in-depth analysis that needs to be done. So thank you very much, and I look forward to the discussion we will have.
Thank you very much to all the speakers. That's really great. So I think in this question and answer period, people will raise their hands. Is that correct, Tricia? And we will also be monitoring questions in the chat. So does anybody have a question for anyone to start off with? I have one for, actually I have one for David. So David, if you think about remote sensing and what remote sensing can now measure, you know, a lot of, there's new capabilities. Do you see that there are gaps in our ability to measure things at a global scale? And if so, what are those gaps that you think we need technical innovations for?
Well, that's a very complicated question because we can only measure a very few things from space. It's just the list is longer than it was a decade ago. And we're limited to measuring things that interact with the electromagnetic spectrum in an interpretable way. That means we tend to see the surface of things, the top of the canopy, the fully illuminated upper ocean, bare soil, and we don't typically see very deep into anything. But you've asked the question in the right way, and the question is there anything that we could measure where could is really the operative word? That is, there's a physical basis for doing it that we're not measuring now or in the relatively near-term future. And there is a list of things that we would like to measure remotely, either for the first time or better. Many of them have to do with structural characteristics of vegetation. So right now we have on orbit the JEDI LiDAR, which is a remarkable instrument, but it samples the world. It gives us data in places where we don't have data from aircraft or people on the ground, but it doesn't give us wall-to-wall data. So while it gives us very large-scale patterns of biomass and forest structure, it doesn't give us very good continental or subcontinental details on forest demography, disturbance, and gap dynamics. We're starting to get very high resolution, panchromatic and multispectral images of the world. There's a wonderful paper in Nature cataloging basically every tree in semi-arid Africa one by one using planet high resolution data. The potential of that kind of technology that is imaging plants one by one for large areas of the world. It's not a gap, but it's something that we've barely begun to extrapolate. I would kind of turn your question around, though, and point out an observation that a colleague of mine who works for NatureServe made to me. He said that in North America, NatureServe has millions of plot-based data, on the order of 4 to 6 million plots, of which 2 to 4% have ever been resampled. Yet we live in a world which many of our speakers have emphasized is changing very, very rapidly. And one of the big gaps really is in the commitment to making some of our diversity-oriented observations on timescales that are relevant to the pace of change. And so it may be that what we need is not more and more observations from space. We've already kind of got something like the basic ecosystem science suite: plant functional traits, canopy height, gas exchange rates, evapotranspiration. But now what we need is to cast those measurements in the time domain and use them to do continental scale process studies, looking at how they change and vary with changing climate, with the interannual variability of the climate system, and with other influences, for example, invasive species and land use and disturbance changes. So to me, in some ways, the big gap is that we didn't start making these measurements 30 or 40 years ago when we first started measuring greenness.
Great. Thank you very much. Ines?
Hi, thank you, David. I have a question following what you just said. Do you have any good examples of people who are doing what you just told us we need, working with this high resolution data and bringing it up to a larger scale quantification of patterns or trends, or even forecasting future dynamics?
Well, are you asking me to name names? I mean, I know.
Yes, I do.
So Janine's group, she's online still, I hope. Janine's group has done scale crossing measurements using spectroscopy. The NASA Above team working in Alaska and Canada has done fantastic work linking in situ centimeter scale drone observations, meter or 5 or 10 meter scale airborne observations, and satellite observations together. The work in Africa was led by Jim Tucker and his team at NASA Goddard. So I think that people are starting to use synergistic combinations of measurements made at different time and space scales. I will just say again that in many cases, the big gaps are in the time domain. For example, we know that plant functional traits, chlorophyll, nitrogen, vary dramatically over the course of the growing season, but almost all of our in situ and remote observations of plant functional traits are made once. I mean, in the case of the TRI database, like once per decade or once per 40 years for a given site. But the image that I showed of the California coastal area shows that those traits change on a weekly basis within an ecosystem and across landscapes. So high resolution is wonderful, and it's very attractive because the technology from drones and the planet constellation and other commercial vendors are available, but ecosystems are dynamic and space only captures part of that dynamism.
Okay, thanks, David. I think we should go on. Jack, do you have a question?
Yes. So I have a question for Thomas. Thomas, you presented this fascinating results, the changes from this in alpha and beta biodiversity due to urbanization. And could you elaborate the mechanism behind that?
The mechanism between the species area relationship?
The species, the changes in different types of biodiversity.
Yeah, so I think the relationship that you have an increase in biodiversity with city size could be related to more resources, human innovation, and better governance and management. I think that, but that needs to be sort of studied more in detail. But I think that's probably the mechanism behind that relationship.
Thank you so much.
So it's sort of a human induced factor there as well.
Okay, there's a question in the chat for Sydney. So Sydney, what other synergies do you know of provided by a network of networks approach?
Yeah, so I actually prepared some slides along those lines in case if we are able to share them, but I don't need to if not. But there's one nice synergy that we've seen in work also with LTER and NEON and across OBFS, the Organization for Biological Field Studies, was that we at an LTER synthesis working group, we were thinking about how you could actually start to synthesize some of these data sets to then do these kind of continental scale analyses. And one of the challenges to any kind of data synthesis is that oftentimes groups will reinvent the wheel. So they'll go ahead and like do all of the data wrangling for their particular analysis and data cleaning and QA/QC, but maybe not pass that on to the next group, which is kind of a key piece here. And this is a lot of what Martin was talking about in his talk. And so kind of to have that kind of data harmonization, if we can come up with these kind of community data standards, Emily also spoke about this with the genomic work. We really need community data standards. And so with that network of network approach, what we did is we built a community data standard for community ecology data through the LTER synthesis working group. But then we went ahead and we've now applied that to also the NEON data. So now we have this kind of way that we can seamlessly integrate NEON and LTER community ecology biodiversity data because they're all in the same format. And so if we can agree upon formats to use across these different networks, then it makes the harmonization of the data much easier and will enable much more synthesis of all of the data.
Great, thank you. Yeah, it's relevant to your talk, Martin. Yeah, very true. So I have just a quick question for Emily. And so Emily, in terms of the metagenomic analysis and being able to define traits, can you then go to these higher levels of organization and see if your trait prediction actually fits with measurements that are non-microbial? Is there a correlation, or is there things that still need to be evaluated to figure out what those correlations might be?
Yeah, thanks, Louise. That's a great question. So we're only at the very basics of doing metabolic modeling for individual organisms. And so being able to have that community level metabolic modeling is still, I view as a gap. I think the emphasis would be that trying to identify what that community level modeling could be, and making sure that you have all of the appropriate measurements and data at the outset, as opposed to doing, sampling of a particular environment, but then not identifying maybe some rare organisms within that community that could be having some really important crosstalk across, the metabolic space that they operate in. So I'd say that we're probably still at the very beginning of just cataloging what's out there and making sure that the data can be more broadly accessible to then feed into newer models.
Thank you. There's a question from the chat from Janine. Oh, wait, where'd that go? Janine Cavender-Bears. So for plant
it's just a comment. It's in response to Jack's question.
Okay. Yeah. Okay. All right. those are all comments. Anybody else have any additional questions, or does anyone want to bring their comments out for discussion?
I have a question.
Okay, Stephanie.
Yeah, so I'm sitting here thinking about, what's missing, and I think we haven't talked about any of the ways that we can scale information about animals, for example, and everybody can, think of their own system. Freshwater, is where I work, and there's some big challenges with that. But I'm wondering, just thinking about animals for a moment, are we sort of, in terms of scaling using remote sensing or other methods, are we kind of still at the point where we have to do, modeling based on what we know about associations between animals and the ecosystems we can measure from space? Or are there some more creative ways that we can scale information about animal distribution and movement? Is there some bycatch from some of these other tools that I'm not thinking about? Is there sort of bycatch of animal microbiomes in the microbiome data that some of you have worked with? I don't know. I would just love to hear what some of you think about the potential for scaling for animal distribution and abundance and movement.
Stephanie, one of our major initiatives at JPL in partnership with colleagues in Germany is to enable animal tracking from space using very, very small tags on the animal itself. And what that does is it enables continental to global scale tracking of migration, as well as local scale tracking of animal movement. The other approach that we've been using for incorporating higher trophic levels and microorganisms is to coordinate remote sensing with environmental DNA sampling. And that has revealed patterns of landscape associations that probably would have popped out eventually, but perhaps not as clearly as they have. So I think that there's tremendous ferment in this area of linking technology to the study of animals, and not as we've done it historically through habitat, but doing it in more direct ways. On the one hand, very large scale using tracking, and on the other hand, going directly to the organism level using environmental DNA in conjunction with correlates from space-based measurements.
Great. Jack, do you have a question?
Yeah, I have a question for Martin. Martin, you have an excellent center there in Germany. You mentioned a number of limitations. What's your plan for the future to address those limitations besides you talked about? Thank you.
Yeah, that's a good question. Given that we get the funding we envision for the next years, that's of course the basis for that. I mean, I can't talk about my colleagues. I'm not above them to tell them what they should do research on. But I guess what we are working on is, for example, for plant traits, finding ways to make these different databases interoperable. So really going beyond ontologies, which maybe are not necessarily always useful, at least not at the end user side. We will also mobilize a lot of knowledge from floras and other not yet digitized data, and with the help of artificial intelligence. There's a huge call ongoing in Germany, which will lead to a lot of projects using AI for certain things. Beside that, talking a lot to stakeholders to find ways to bring in these topics into calls of next funding rounds to address that then with the next round of funding. That's how my work is working. Yeah.
Thank you so much.
Okay, Scott.
just a quick follow-up to your question about animal databases and so on that Dave gave an excellent answer to. I think we definitely want to incorporate that in this report. And there are some very rich data sets now, animal movement, migration data sets in MoveBank and in other places, very large camera trap networks and so on. So there's really tremendous potential for using those data sets together with all these rich remote sensing data sets, not specifically, but including these remote sensing data sets that we now have access to, and canopy structure and traits and so on. So I think there's great opportunity there, and we'll be sure not to overlook that.
Great. We have time for one more question.
Yeah, I have a question.
Okay.
Yeah, excellent. Really enjoyed all these talks. Excuse me, I have an echo here, so I'm going to turn off. Hopefully you can still hear me. So we heard a lot of talks today about we're in a golden age of data and this rich explosion of data. And these are wonderful macroscopes and microscopes. We can now kind of view the tapestry of life at just incredible scales. And we still have issues. There are a lot of gaps. And then kind of building on what Stephanie was asking about, what is missing here? I keep being haunted by this quote by the Nobel laureate Sidney Brenner that states that biological research is in crisis. Technology gives us the tools and the access to measure and observe organisms at all scale, but we are drowning in a sea of data, and we are searching and thirsting for some theoretical framework by which to understand and simplify it. And so I guess my question is a general one, for the different speakers. What is the role of theory, and do you need theory to help organize your data? And if you did need theory or could access theory, what would it be and what would it tell you? I know that's a big question, but in thinking about this report, these are the types of things that I think about and I'm interested in.
Great question.
Dave, what about you?
Well, I think it is a great question, and I would say that we use theory to frame our observing priorities, and we attempt to interpret data, at least with reference to theory, although often there's a gap between what we can see in the data and what theory predicts. So I have a number of theoreticians in our carbon cycle and ecosystems group at JPL, and we're constantly trying to make the bridge between relevant theory. We rely very heavily on stoichiometric and phylogenetic approaches to the observations that we can make. And this is something I've spent many hours talking to Janine about. With remote sensing, we tend to think about 30-meter pixels, which are not exactly organisms. And yet we'd like to ultimately interpret those spatial and temporal patterns in terms of underlying biological and biophysical process. And that connection has to be from theory. Empirically, there are too many gaps between the observation and the creatures that are making those patterns at 30 meters in some collective way. There's really no way to approach the interpretation other than either endless regression analyses or theory. And if you want to point to gaps, there are many scale gaps in our theory, as you well know.
Great, thank you. Thank you all, the panelists today. It was a great discussion. I think we now need to wrap up, and Jack will do that for us. And again, thank you for your presentations and efforts and interactions.
Yeah, thank you so much,
Oh.
Louis, and you might speak on. Yeah, thank you so much, Louis, all the speakers and committee and the audience for the excellent presentations and discussion. I have learned so much, and due to the time limitation, I will just highlight a few points. And one is that much progress has been made from Janet's keynote presentation. She connected two grand challenges of our times together: global change and biodiversity loss. The points that she made would really help to effectively forecast the impact of global change so that we can prepare for preventing and reducing biodiversity loss, even to restore biodiversity in the future. They will highlight the golden age of remote sensing. The various remote sensing data can help generate information and useful knowledge across multiple scales. Emily talked about the golden age of genomes. I think that's really exciting and issues. And Nova talk about integration of microbiology and epidemiology and biogeography using citizen science approach. I think that's something that is becoming more and more popular. And Sydney explore drivers of small mammal diversity using neon data. And Thomas really highlights some exciting results about the patterns of change in biodiversity under urbanization and globalization as well. The second main issues that I would like to touch upon is the challenges and limitations that the speakers and talk about, especially the data availability and the data synthesis. And for example, Martin mentioned several points in this regard. And also he mentioned the no funding or not enough funding to maintain the data or to make the data useful for other people to use. And also how to use the data effectively require really in-depth teaching and training. Right? And the final point I want to make is the needs and the solutions to the challenges and the limitations. And of course, Martin mentioned several things, and like a more innovative use of AI. And Emily mentioned fill the sample gaps in many regions. And of course, Dave mentioned that not just sampling new plots, but also resampling at the old plots. I think that's very important. And fortunately, there are some long-term sites to resample the same location repeatedly at the regular time interval, but we need much more. And also, Emily talked about resource network, and Cindy talked about networks of ideas, people, and the infrastructure. I think that's very important to implement continental scale biology. And the final point I'd like to make is more integrated theories that Brian and Dave talked about. And I think the other speakers also touched upon. I think that series can guide our research and also really integrate across the different scales and across the different biome and domains and so on and so forth. There are many other important insights presented by the outstanding speakers, but due to the time constraints, I will not do that, highlight those other points and here from the presentation and discussion today. But the good thing is that we will have another webinar tomorrow at 1 o'clock Eastern time. So everyone is welcome. And again, many thanks to everybody, to the speakers and to the moderators and the participant, the audience. And I will looking forward to see you tomorrow. So thank you so much again.